Hi, this video will explain about specifically the process of glycolysis, which is another one part of aerobic respiration. So at the end of the lesson, students should be able to illustrate to explain glycolysis pathway, which is involve conversion of glucose to pyruvate, and describe link reaction, which involve conversion of pyruvate to acetyl coenzyme A. Glycolysis is a splitting. Okay, the word lysis means split, breakdown. One molecule of glucose, which comes to the word glyco. One molecule of glucose is split into two molecules of pyruvate. So as you can see, glucose is 6 carbon molecule, while pyruvate is 3 carbon molecule. So 6 divided by 2 molecules of pyruvate, 3 carbon molecule, through a series of reactions catalyzed by specific enzymes. Glycolysis occurs in the presence or absence of oxygen, regardless of the presence of oxygen. Meaning that if there is not enough oxygen, glycolysis will still occur. Glycolysis occurs in the cytosol or cytoplasm, and the products of glycolysis from one molecule of glucose are pyruvates, 2 molecules of pyruvates, 2 ATPs, 2 NADH plus 2H plus, and 2 water molecules. So glycolysis, we are going to look into the details of this process. There are 10 steps involved in glycolysis, and each steps are catalyzed by a specific enzyme. The glycolysis can be divided, or these 10 steps can be divided into two phases. The first phase is the energy investment phase, which involves step 1 to step 5. During this phase, the energy investment means the ATP is used during this phase, while the second phase is the energy payoff phase. During this phase, the energy is yielded or ATP is formed during steps in between step 6 to steps 10. The first phase, energy investment phase, involves step 1 to step 5. As you can see from the diagram, the glucose is converted to glucose 6-phosphate. This process is a phosphorylation because it involves the addition of phosphate to the glucose. So to use the phosphate, to attach the phosphate to the glucose, one ATP molecule is used. Thus, this process is called phosphorylation. So in another word, we can say that glucose is phosphorylated to glucose 6-phosphate with the help of enzyme hexokinase. The second step is isomerization process, which involves the rearrangement of the atoms in the glucose 6-phosphate to be converted or isomerized into its isomer, the fructose 6-phosphate. So we can say that glucose 6-phosphate is isomerized into fructose 6-phosphate. As you can see, there are a major rearrangement of the molecules in the glucose into fructose, but the phosphate are still maintained at the 6 carbon. The 6 here means the phosphate is attached to the carbon number 6. The third step is another phosphorylation. So fructose 6-phosphate receive another phosphate from the ATP through a phosphorylation process, which will produce fructose 1,6-bisphosphate. So The phosphate, another phosphate from the ATP is bound to the first carbon of fructose 6-phosphate. So, fructose 1,6-bisphosphate is produced from the process of phosphorylation. So, as we can see, phosphorylation involves the use of ATP. So, this is the second ATP that is used during energy investment phase. The enzyme involved in this phosphorylation process is phosphofructokinase because the substrate is already a fructose 6-phosphate. Students should memorize these two names of enzyme, hexokinase for the first step and phosphofructokinase for the third step. Next, the fourth step is the splitting, the breakdown of fructose 1,6-biphosphate into two molecules of 3 carbon. So the 6 carbon molecule, fructose 1,6-biphosphate, will be split into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. You can memorize the abbreviation of the molecule. Except for G3P, you have to know that it is named as glyceraldehyde 3-phosphate because the fifth step is the isomerization. In order to continue to the sixth step of glycolysis, DHAP cannot continue to the next step, to the step number 6. Only glyceraldehyde 3-phosphate can continue to the sixth step. So, DHAP should be isomerized into glyceraldehyde 3-phosphate. So this is the end of energy investment phase. We are going into the energy payoff phase. The 6 steps which involve glyceraldehyde 3-phosphate. So since we have the DHAP that is isomerized or converted into a G3P, so in this step, starting from this step, we have 2 molecules involved. So, whenever you are going to mention about the 6 steps, don't forget that it involves 2 molecules of glyceraldehyde 3-phosphate. Step number 6 shows that glyceraldehyde 3-phosphate is oxidized, meaning that it will lose, it donates hydrogen away. So hydrogen is removed from the G3P. That is why G3P is said to be oxidized. So the hydrogen that is removed from the glyceraldehyde 3-phosphate will be received by the NAD+. This is a redox reaction, which means NAD+ is reduced to NADH plus H+. So the oxidized G3P will receive phosphate inorganic from the cytoplasm. Yes, phosphate inorganic means that the phosphate group is not attached to any organic molecule. So the phosphate is a free phosphate in the cytoplasm, will be attached to the oxidized molecule, oxidized G3P, and thus it will form 1,3-biphosphoglycerate. Students should memorize the name 1,3-biphosphoglycerate. So step 6 involves first oxidation of the G3P and then the phosphorylation. Phosphorylation is when the phosphate inorganic is attached to the resulting molecule during the oxidation. G3P is oxidized and release the hydrogen to the NAD+, and then receive phosphate inorganic and converted into 1,3-biphosphoglycerate. So step 6 is about oxidation and phosphorylation. Next, step 7 involves substrate level phosphorylation. So whenever you come across substrate level phosphorylation, meaning there is a production of ATP. 1,3-biphosphoglycerate receive phosphate group from the ATP. 1,3-biphosphoglycerate will donate or remove phosphate group and will be received by the ADP. So ADP will be phosphorylated to form ATP. So since from the step 6 it involves 2G3P, so everything must involve 2 molecules also. So at step 7, 2 ATP is produced from the phosphorylation, removal of phosphate from the 1,3-biphosphoglycerate. Since 1,3-biphosphoglycerate remove or donate the phosphate group, it will be converted to 3-phosphoglycerate. Then 3-phosphoglycerate will undergo isomerization during step 8. The isomerization involves the rearrangement of the phosphate group from carbon number 3 to carbon number 2. 3-phosphoglycerate is isomerized to 2-phosphoglycerate in step 8. Next, during step 9, 2-phosphoglycerate undergo removal of water. Thus, it will be converted to phosphoenol pyruvate. Since we still have a phosphate in the molecule, in the substrate, it comes to the end of the stage, which involves another substrate level phosphorylation. So phosphate group from the phosphoenol pyruvate will be removed and phosphorylate the ADP to ATP, meaning that ATP is produced. So this is what we call as substrate level phosphorylation. So the phosphoenol pyruvate release the phosphate group and converted to pyruvate, which is the end product of the glycolysis. So another 2 ATP is produced. To sum up, we will have 4 ATP during energy payoff phase. Okay, the explanation during each step. During phosphorylation, the enzyme hexokinase transfer a phosphate group from ATP to the glucose, forming glucose 6-phosphate. The phosphorylation helps to make the glucose more chemically reactive, which is a very important characteristic to continue with the process. A more chemically reactive molecule can make the molecule becomes easier to be manipulated, because after this the molecules need to be isomerized. So glucose 6-phosphate is converted into its isomer, fructose 6-phosphate. Another phosphorylation in step 3, ATP donates phosphate group to fructose 6-phosphate, catalyzed by enzyme phosphofructokinase. So during energy investment phase, 2 ATP molecules has been used. Fructose 1,6-biphosphate split into 3 carbon sugars, the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The dihydroxyacetone phosphate is converted to its isomer glyceraldehyde 3-phosphate to continue with the steps of glycolysis. Because Only glyceraldehyde 3-phosphate can continue, okay, because the enzyme in step 6 can only feed the glyceraldehyde 3-phosphate as its substrate. Step 6 involves oxidation and phosphorylation. So the glyceraldehyde 3-phosphate oxidized and then phosphorylated by receiving phosphate inorganic to form 1,3-biphosphoglycerate. During this process, an ADH is produced. So the energy released from this exergonic redox reaction will be used by the oxidized substrate to attach phosphate inorganic to the oxidized molecule. Step 7 involves substrate level phosphorylation, which is 1,3-biphosphoglycerate converted to 3-phosphoglycerate by removing one phosphate group. So the phosphate group from the 1,3-bisphosphoglycerate is transferred to ADP, forming ATP. So this is the process of substrate level phosphorylation, which produce 2 ATP because it involves 2 glyceraldehyde 3-phosphate. During step 8, isomerization, the 3-phosphoglycerate is isomerized or rearranged, forming 2-phosphoglycerate. And step 6 involves removal of water from 2-phosphoglycerate, which produce double bond and produce phosphoenol pyruvate. Step 6 involves phosphoenol pyruvate transferring its phosphate group to ADP, forming ATP, which is the substrate level phosphorylation, and the last product is pyruvate. So, as a summary, during phase 1, energy investment phase, 2 ATP is used, while in phase 2, energy payoff phase, 4 ATP is formed. So the total amount of ATP is 4, but the net ni maksudnya bersih, amount bersih. Amount of ATP is 2. We produce 4, we pay the debt. Kita bayar hutang yang kita guna tadi, maknanya kita tolak 2. So bersihnya kita dapat 2. Another summary, from 1 molecule of glucose, we will produce 2 pyruvate, 2 ATP, 2 NADH plus 2 NADH plus 2H plus, and 2 water molecule during step 9. After glycolysis, pyruvate will proceed with link reaction, while NADH plus H plus will proceed with the oxidative phosphorylation. The NADH will transport the hydrogen and the electron that it received from the glycolysis to oxidative phosphorylation. That's why NADH is considered as electron carrier. After glycolysis, when oxygen is present, and ADH, which is the electron carrier, will transport the electron into the mitochondria. So the electron will be carried or transported to the ETC, which is at the inner membrane of mitochondria. The electron will be used to produce ATP through oxidative phosphorylation. If oxygen is absent, for example during vigorous training, maknanya kita exercise yang berat lah, exercise berat, so oxygen is not sufficient. The NADH will become reducing agent in the cytoplasm and produce lactate or ethanol. This will be learned in subtopic fermentation. Question 1. In which two steps are ATPs being used and why? Step 1 and step 3. Why? To increase the energy, the reactivity of the glucose. So glucose becomes chemically reactive, or the substrate become chemically reactive. In which step is NADH plus H plus produced? And state the function of NADH plus H plus. Do you remember? NADH plus H plus is produced at step 6, and the function is as oxidizing agent. Another function to be mentioned is NADH as electron carrier. How many molecules of NADH and pyruvate are produced from one molecule of 1,3-biphosphoglycerate? So the answer should be one molecule for NADH plus H plus and one molecule of 1,3-biphosphoglycerate. One molecule of pyruvate. One NADH and one pyruvate, because only one molecule of 1,3-biphosphoglycerate. Link reaction. Pyruvate is converted into acetyl-CoA. It occurs in the mitochondria during the transport of pyruvate into the mitochondria. This acetyl-CoA will be used in the Krebs cycle. So link reaction involve oxidative decarboxylation of pyruvate. Oxidative means it involves the redox reaction. Decarboxylation means it will lose a carboxyl group in the form of carbon dioxide. So there are 3 steps in link reaction. First, decarboxylation of pyruvate. Removal of the carboxyl group from the pyruvate, forming carbon dioxide and 2 carbon fragments. Okay, these 2 carbon fragments then will undergo oxidation, forming acetate. Acetate is 2 carbon molecule, so no more removal of carbon. So the 2 carbon molecule oxidize, right? It undergo oxidation by transferring electron and hydrogen ion to the NAD+. So NAD+ will be reduced into NADH plus H+. Then acetate will receive coenzyme A. Coenzyme A to form acetyl-CoA. Then acetyl-CoA will enter Krebs cycle. So as a summary, the products of link reaction involve 2 NADH, 2 acetyl-CoA and 2 carbon dioxide. Kenapa dua semua ni? Because we have 2 pyruvate. 2 pyruvate from the glycolysis. So, during aerobic respiration, after link reaction, acetyl-CoA will proceed with Krebs cycle, while NADH plus H plus will proceed with oxidative phosphorylation. The function of NADH plus H plus still as an electron carrier. Question 4. What is the net ATP production for glycolysis and link reaction of 6 molecules of glucose. So the keyword is 6 molecules of glucose. So for 1 molecule of glucose, we should get 2 net ATP. So for 6 molecules of glucose, we should get 12 ATP. For link reaction, we do not produce any ATP. Okay, for link reaction, it is 0 ATP. Okay, thank you.